Yeah, good morning. Welcome you all to the next lecture on Organic Chemistry of Life, Principles and Perspectives. In the previous class, we have tried to look at the phosphatases and of course ribonucleotide reductase, etc. Then we just started towards the end of the class the nitrite reductase. So, we continue with some nitrite reductase. The reason that I have taken this reductase afterwards is this is a heme based one, the one which I have talked about, the ribonucleotide reductase, is non-heme based place. Even the phosphatase, purple acid phosphatase, was also non-heme based one. It is only that the classification, otherwise there is no other problem. Okay. Now, let us look at once again I will explain the couple of things that I have done already in the previous class on the nitrite reductase. Nitrite reductase is an enzyme which converts the nitrite group into nitric oxide kind of a moiety NO2 to NO. So, obviously what it requires NO2 to NO, it requires one of the O to nitrogen bond should be taken out. So, how do you take out? We have already seen earlier. So, whenever you want to take out, you add the electrons into the anti-bonding orbital and then you add some protons, that O will go as a H2O, or you have a substrate, it can add to that too. So, very similar kind of analogy will work out here. So, which means this enzyme requires some the substrate has to bind, substrate has to reduce. So, therefore, some electron transfer should take place and then finally you break one of the NO bond, etc. So, therefore, in this enzyme we could see there are two types of heme based centers are there, which is called the C type, heme C and heme D, okay. And there are two each of these because there are two polypeptide chains in that. So, therefore, you have a dimeric kind of a protein. Okay, there are different forms of these enzymes are there and they do different levels of the reductions. Some of them do from nitric oxide to nitrous, NO2 to NO, and some of them can even do from NO2 to ammonia as well. We will study both of these cases as we go along in this particular class. The one which does from NO2 to NO is a kind of a thing which is known as ferrocytochrome C551 oxidoreductase. Okay. So, in this case you have a reduced heme of the D type which will bind a nitrite group and then converts into that. So, that means the D part of the heme is catalytic type and the C heme is the one which is involved in transferring the electron. Okay. So, therefore, cytochrome C nitrite reductase CCNIR is a multi-heme enzyme that converts nitrite to ammonia, which we will take up after completing the NO2 to NO kind of an enzyme. So, as you can see below here, there are two types of heme C and then heme D. What is the difference? You can see that there is oxidized part of that. That means some of the portion of the skeleton of the porphyrin is oxidized in this particular case, as you can see that non-oxidized and then oxidized. So, what will this differ? Obviously, this will differ on the way that its redox potential is lying. Okay. So, the redox potentials of the C versus the D are going to be different, and as you can see the C is involved in electron transfer while D is involved in the catalysis, or where the NO2 binds and then further reduction or catalysis takes place in all this. So, let us look at more details of this enzyme. In the more details of this enzyme, so as I said that this CD1 nitrite reductase catalyzes the conversion of NO2 to NO in the denitrifying bacteria. Okay. So, if you look at, as I mentioned in the previous slide, that the D heme is the center where the reaction occurs or where the NO2 binds. Now, if you look at that, the same thing here, as you can see, this is the ion center, this is the kind of heme, and this nitrite group binds. Let me bring one point clear to you in terms of the kind of a heme that we have. Let us go back to the previous slide, and sorry, this one, yeah, here you can see that. So, there is a kind of a heme C type, heme D type, oxidative type, and therefore, the redox potentials vary. Yes. So, therefore, that is an important point to be noted. So, therefore, that kind of a center which is D is very well suited for binding to the NO2. Now, what is to be noted here is a very interesting thing is this NO2 has some interaction or secondary interactions with this kind of histidine, with this histidine. In this particular enzyme, this is called histidine 327 and histidine 369. So, therefore, these two histidines are absolutely important. They have a very important role to play. How do we know? Whenever we say that a particular amino acid residue plays an important role, what should be the trick? What should be the tact? Trick and tact in this is you replace that amino acid residue by a different amino acid. What is this process called? Mutation. So, you mutate the protein or enzyme at that particular center to a different amino acid which does not have the same side chain. Therefore, that kind of a process is called mutation, and when you do the mutation in this particular case either at histidine 327 or at histidine 369 or at both these, what they find is the outcome of this is there is no product that is coming out of this particular enzyme. The particular enzyme does not give the product. Actually, it is not that the reaction does not occur, reaction occurs to reasonable extent, but the product does not dissociate, product does not come out of this particular system. So, therefore, whenever you mutate and change these histidines, what you will have essential is reaction happens, but catalytic cycle does not complete because the NO does not dissociate. That is a very important point. Okay, and one more thing that could be realized when you do the mutation is that there are certain things. You can see there is one tyrosine moiety, tyrosine 10. So, therefore, this tyrosine 10 also get perturbed when the mutations have taken place, and also we need to see within 1 to 2 nanometer vicinity the protein environment is also important. So, what happens when you do the mutations? There is some new side chain at the active site, or some of the tyrosine things will go away, the tyrosine slips out of this region and tyrosine density will not be seen that much, and a lot of topological changes also occur in the vicinity of this. So, therefore, what we can say is within 1 to 2 nanometer area surrounding this particular region is of the protein residues are very important, because you have to have electron transfer, you have to have a reduction process, removal of the oxygen too. So, therefore, the crucial thing. Of course, the catalytic activity comes at D side and the electron transfer comes from the C type, and this can be seen very clearly in the next slide. Similarly, you can see these things are involved over there. Okay, so let us look at some structural aspects of this cytochrome CD1 or nitrate reductase enzyme CD1 in its oxidized form. In the oxidized form of the CD1, the mainly helical C type cytochrome C domain binding the one domain that is which is the below. You have two types of domains, the domain 1 which is the D1 which is over there, and then you have the cytochrome C type which is coming from, it is the cytochrome C which is involved on the top, and the D is which is over here, and that is how it is involved. Okay, and if you now look at the close proximity, how do you find which is cytochrome C type, one is D type? You look at the binding. The binding of histidine 17 and histidine 16 as axial ligands, that will qualify the cytochrome C, and in the other case it will qualify this cytochrome D will be have histidine 200 and histidine 25. Now, let us bring up the mutation. The mutation is one of the tyrosine is converted to the cysteine, okay, a serine. Okay. One of the tyrosine is converted to serine Y25S. In this variant, it is essentially like that of the wild type, but there are some differences in the vicinity of the mutated residue. Okay. The C type does not get perturbed, both the hemes are both the histidines are there, but what get perturbed is the D type. The D type 1 is ligated to histidine 200, but one of the ligation is lost, the other ligation which is tyrosine 25 is lost, and in this position a sulfate ion binds and anion binds. So, this also gives a clue that this is very well suited, the cytochrome D type is well suited for binding to the anions or to the substrates, and because earlier it was the tyrosine 25. So, therefore, if you modify this tyrosine 25, then obviously that is not going to bind, and therefore, that can be seen very well. And this is the position where the anion or other substrates can bind, and can be very nicely seen from the crystal structure. Sulfate binds over there. That gives an explanation for how it binds. Now, let us look at the catalysis of this. Okay, the catalysis of this at the D center. So, let us look at the D center here and in the reduced form, which is the iron 2 form, and in this is the center where the NO2 substrate binds, and this substrate has an interaction with the neighboring nearby histidines, as I showed you on the earlier slide, and therefore, this is what brings recognition for the substrate binding. So, substrate binding recognition, as I told you earlier, requires secondary interactions, in this particular case these two. And additionally, these two histidines have got a lot of importance, as I told earlier. When you mutate these, the reaction goes, but reaction does not complete because the NO does not dissociate. Now, this NO2 is activated by the iron 2 obviously, and the electron transfer taking place into that in presence of the two protons will break down this to the water and the Fe3NO, and this could be in a resonance structure with the Fe2NO with the radical. And this is the species which has to dissociate to give back the ion, but the evidences are not built for this, as I have written on this side, and this iron 3 is not the active form, then there has to be necessarily an electron transfer. That electron transfer can come from the cytochrome C part of it, and then can go to the iron 2. So, at this stage the species is referred as the FeNO, which is called FeNO6, which could be either in this form or in this form. If it is in this form, the dissociation has to occur, and the dissociation process is not being proven to this, but the reduction of this is required. On the other hand, if it goes via the Fe2 with the nitrosyl radical type, then the further electron transfer should bring a different species called FeNO7. Again, this should dissociate, and no evidences have been shown completely for either of these, but there is a possibility. Since this can undergo transformation to the other, there is a possibility that either this process can happen, though this process has not been shown yet. So, only when the NO is dissociated, the catalytic cycle can get reversed here, okay, and that will take out the NO and then brings in. So, either it can go through this process, or it can go through the iron 3 and then further reduced, losing the NO and then losing the going back to this one. Okay. So, this is also a possible thing, because the possibility for this is because the reduced one iron will have a more of strong pi back bonding stabilization, whereas iron 3 will have much less of such kind of a stabilization. So, there is a good reason to believe that it will break down with the iron 3 and then reduce to this too. Okay, let us look at the other enzyme which reduces the O2 not just to the NO, but to the ammonia level. So, obviously, in the process it has to go through NO and then go through this, but the very minimal things are known. All this is written here. I will be explaining in the catalytic cycle all of these. So, therefore, we do not need to worry. You can read after looking into the catalytic cycle these ones. Let us look at some characteristics of this nitrite reductase, and this can also do sulfite reduction too. Now, first let us look at the reactions. Look at the reaction of nitrite. So, NO2 minus plus 6 electrons plus 7 protons will give ammonia plus 2H2O. That means, here in previous example NO2 to NO, therefore, only one oxygen goes as the water, but in this case NO2 to ammonia, both the oxygen must go as water and require more electron reduction and more protons, because the ammonia nitrogen will take 3 protons, 2 waters will take 4 protons, totally 7 protons, and the NO2 minus requires only 6 electrons to reduce completely, to reduce the oxygen parts to go to this water, and then nitrogen part to get to the N3 minus. Okay. So, therefore, that is where you have. Okay. So, the total counting should be perfect, and you can see that. So, 6 minus 1 minus 7 plus. So, that is neutral. Now, if you go take the sulfite case is also very similar, and 6 electrons 7 protons, it will give Hs minus plus 3 H2O. So, that is 7 protons. So, you can see that. So, it is very much similar. In this case you have a different kind of a heme center. This heme center is referred as a cyroheme. This is a cyroheme. This heme has got a heme, has got an iron sulfur cluster, both of these are connected together. So, earlier we studied for electron transfer, you have either a heme kind of a protein or you have a iron sulfur kind of protein. Here is a case where both the iron sulfur part and the heme part are connected. It's connected through this thio bridge. The iron of the heme to the iron or the iron sulfur protein or iron sulfur cluster is connected through the this kind of a thing. So, this is referred as a cyroheme. So, you know this the normal hemes we have looked at earlier will have plus 50 to plus 300, whereas the cyroheme, depending upon the groups changed here as well as the protein that is surrounding, this can go between minus 50 to minus 300. So, why do you require such a? Because you are going to reduce the NO2 minus to ammonia. So, you need a very strong reducing center, and that is why this enzyme has taken, nature has decided to put a different kind of a reductive center than the normal one, and that is what you can see that. So, is it understandable? Now, we have seen earlier iron sulfur cluster, then heme is a combined one, you can see that, and this is because you require a very high negative potential, and this kind of things will show more negative potential minus 50 to minus 30. Now, this protein will also have, as a function of pH, the redox potential vary, and as you can see the slope, if you look at the slope and the behavior of this, you can definitely say the electron transfer is coupled with the proton transfer. So, both the electron transfer and proton transfer events are coupled together, that you will get from this particular kind of. This kind of plots are called Porbok's plots, pH versus redox potentials of this. So, the sulfide reduction reaction, nitrite reduction reaction to ammonia requires a strongly reducing center, therefore, a cyroheme is being utilized in this particular case. Now, let us look at the proposed mechanism. The reason is that many of these steps have not been experimentally shown, but some DFT computational calculations have supported, have proven, have brought in this one. So, based on that this particular scheme is given. Okay, first of all we should say that the not the oxidized form is the active center, the reduced form, that means iron 2 center. So, the iron 2 center, so the The resting form of the iron 3 is not the one and it is the iron 2 center is reactive, and at this stage that you have a NO2 binding. So, NO2 binds here, and this binding of NO2 is facilitated by the neighboring groups. One is the histidine and the tyrosine, as you can see over here. So, this is one of the species, and then the protonation to this, and since this is bound by this will pull out the NO bond, it will stretch the NO bond further because of these secondary interactions with the histidine and tyrosine, and that favors the breakage of this O in presence of the two protons as a water, and then what you get, a nitrosyl kind of a species, and this species has a number given FeNO6. So, just nothing to do with the 6 or 6 plus anything, is a species 6 which is computationally derived. Now, this with further reduction with one electron, and that electron gets into this particular the part, and therefore, the binding of the FeNO is now become bent, and this is again being computed by the DFT computation, and this species is called FeNO6. And then one more electron will further add to this, and that will make the further in the reduced form of it, and at this stage the protonation you can see FeNO, and then one more the two more electrons will add into this, and the protonation will take place, and at this stage you have the 3 protonation, the water obviously will go out because of these 2 protons and electron. So, these 2 protons and electron you require. So, you have the water is last, and then now this further this species one more electron reduction will perfectly get the NH3, and in presence of the NO2 this NH3 can be taken out as NH4 plus ion. Now, you can see that there is the electron counts, you could see that from ion 2 plus there is 1 electron added over here, 1 more electron added here, 2 more electrons added here, that is 1 plus 1 2 plus 2 4 plus 1 5 plus 1 6. So, totally you have a 6 electrons, and you see there are 2 protons added over here, and 1 proton, that 3 added plus 2 more 5 added plus 2 more 7 added. So, you have totally 7 electrons added. So, many of these species are shown by the computation DFT computational studies, as you can see that whole thing. So, the active form of this one is not the iron 3, it is the iron 2. So, therefore, the cycle can continue with it, okay, and the reduction can take place back to that thing. So, now let us go back to the earlier slide where I said, so all these points that you can see, so that based on the density functional theory, this has been very well worked out, the species kind of thing. Of course, the cleavage requires the electrons to be pumped in, protons to be there, and a pulling of the NO bond by the neighboring groups, and the first one happens, and that is where FeNO6 species has been shown, and that is what we have explained to you. Then it goes to the FeNO7, then goes to the FeNO8, these kind of a further reduction followed by this one. So, totally, so this enzyme, lot of the species have been worked out, have been experimentally shown, but DFT computations have clearly favored these kind of a species. So, species 6 type, 7 type, 8 type, and the reactions become very fast after this. So, the active form is +2. Totally, you have to have a 6 electron reduction. The activity comes from the ion 2. Okay. So, in the kind of a reductase based on the heme, we have looked at 2 aspects, NO2 to NO stage, then we have looked at the NO2 to ammonia stage, there are 2. So, to have the NO2 to NO, then we have a C type and D hemes, whereas to have NO2 to NH3 type, we have another one which is called cyroheme. Cyroheme means a heme which is connected with a iron sulfur cluster through a thiolate bridge. So, is that clear? Everything is there. Now, this brings to the end of the different types of the enzymes that we are trying to look at, which are all based on the iron. Some of them are based on the iron heme type, some of them are based on the non-heme type. So, let us look at the summary of what I have done in the past 8 to 10 classes or 7 to 8 classes or so, which I have consumed or utilized for the iron enzymes, is a kind of a recapitulation. Let us look at or a summary. Okay, so the iron enzymes I have talked to you about the oxygen transport one. In the oxygen transport one, I have talked to you the heme based hemoglobin, myoglobin. Hemoglobin does the transport, myoglobin does the storage, etc. Then I have also looked at some other cases, the molluscus and other kinds of layers species where non-heme, which is a hemerythrin, which is di-iron center, hemocyanin, which is di-copper center. Then I compared how the oxygen transport takes place in these versus it takes place in the hemoglobin. Hemoglobin more or less O2 is not reduced much at all, whereas in hemerythrin and hemocyanin they reduce and bind indifferently. All these things have been explained, and they can very nicely be understood from even simple IR spectrum by looking at the OO stretching frequencies we can explain. Followed by that, I have explained to you iron storage and iron transport. Iron transport is by transferrin and iron storage is by ferritin, and in this ferritin again storage iron is brought by transferrin and then transferred to the ferritin, and again you take it out. So, I have explained to you how the ferritin is loaded, how the iron by iron, and how the iron is taken out, mobilized. So, when it is brought, it comes in the form of iron 2 plus, and it is given inside to the iron ferritin, and in the ferritin it gets oxidized and form a ferrihydrite kind of a species with phosphite species and with a bridged oxophosphato kind of a bridge and iron 3. Then when it is to be released, then this iron 3 has to be reduced by some kind of a reducing enzymes, some kind of even electron transfer heme enzymes which will reduce, and then the iron is released. So, these are all I have explained to you. Then I have gone to the electron transfer. There are two types of electron transfer. One is on the heme based cytochrome C. I have fully explained cytochrome classes I have explained, then cytochrome C and how it functions. All these things have been explained. Then there are non-heme ones which are iron sulfur protein, where the iron sulfur proteins are also involved. There are 1 iron 4 thiolates, there is 1 iron sulfur protein called, there are 2 irons and 2 sulfur and then 4 iron 4 sulfurs. In these things, these are the inorganic sulfides. Inorganic sulfide can be taken out by adding the mineral acid as H2 acid comes, and if the inorganic sulfide is not there, no H2S will come, which can be made out. Otherwise you have and then during the electron transfer there is the core of the iron sulfur core, which is a cubane, will contract and expand, contract and expand. That is the kind of thing which brings concomitant changes in the protein structures. So, having explained this transport, storage, electron transfer, etc., then I have taken into the oxygenase properties, means adding oxygen. You can add one oxygen of the O2, or you can add both the oxygens of the O2, which are referred as the monooxygenase or the dioxygenase. And under the monooxygenase, I have explained for the heme case cytochrome P450 completely how the reaction occurs, what is the catalysis, how the catalysis occurs, etc., etc. Species like ferryl oxo species formed, which is very strong. And I also talked to you the O2 activation will take place after the substrate, not before the substrate. Please remember that, because otherwise there will be a great danger. Then I have also explained one another example of the monooxygenase by taking non-heme enzyme, which is called methane monooxygenase, which does methane to methanol, which is not that simple. You need very high pressure, high temperature catalyst if you want to do in a lab, but an enzyme does it so nicely. And such kind of conversion is important because the methane cannot be transported, because methanol can be transported very well. These are energy based materials. Then I have explained to you what is a dioxygenase, the interdiol, extradiol, but I have taken example only for the interdiol, which is protocatechate non-heme based kind of thing. Okay, and so other examples I have not taken purposefully in that, because there are much more severe things are involved in that too. Okay, then I explained to you the class, different kinds of di-iron enzymes, various functions in this, um, and so all those things, classification with the different kinds of di-iron centers. Then I took up the nitrite reductase with the heme, explained to you non-heme ribonucleotide reductase. Both of these are explained. How the two compartments of this protein in ribonucleotide reductase are involved, though there is no substrate binding at the iron center, but it binds in the other subunit of these. These two subunits are interfaced at the C terminus. One is having the iron center, which is called hydrolase, other one is having the sulfhydryl functions, which is involved in the redox. So, together the whole reaction. There is a tyrosine radical which is stabilizing in the ribonucleotide reductase, and in this 2' hydroxy should be converted to 2' deoxy, and that is what is deoxyribonucleoside, because deoxy ribonucleotides are not input to the human system. So, therefore, that is required to be made. So, nitrite to NO, I have also explained nitrite reductase to the nitrite reduction even to the ammonia. Then I have explained prior to that catalase, di-iron center activity, rubrithin, and explained how that binds the peroxide H2O2 and then reduction, the dismutation of the H2O2 to water. I have also explained one another class of enzymes, which is basically hydrolysis, and this gives the phosphate hydrolysis, phosphate ester hydrolysis in this too. So, these are the kinds of things. So, transport, storage, and then oxygenase, oxidation properties, reduction properties, hydrolysis properties, and these are, there are many other details that are involved. One hydrogenases of iron I will explain when it comes to the story of the nickel, and I have purposefully avoided that. Many more things are there. It will be impossible to cover all of the enzymes of the iron in for a particular course. So, therefore, I stop for the iron enzymes at this stage. Thank you very much.